Good morning, madam. My name is Mugilan. My matrix number is 23DBT20F2007. Today, I'm going to show the PCR test, which is the visual simulation, which is visual bacterial identification. So now we're going to see the PCR. So there are six protocols, which is sample preparation, PCR amplification, PCR purification, sequencing preparation, DNA sequencing, sequencing analysis. So first, we're going to do the sample preparation. For that, we have to wear glove. Then we have to obtain the sample using wire loop. So first, we have to touch on the bacterial colony, take the sample, and uh, and transfer the bacterial colony to the microcentrifuge tube. After that, take the yellow micropipette and put some digestive enzyme into the microcentrifuge. Then wait for several hours. After several hours of enzymatic digestion, the sample is ready. So then we have to microcentrifuge tube into the heat inactive the digestive enzymes. So after the after the digestive enzymes are now denatured and inactive, that now we're going to microcentrifuge tube and put on the for that we have to spin down cellular debris for removal from the sample. So click the centrifuge. After that, the unwanted debris have now been spun down. DNA we want is in the supernatant. So we have to transfer the supernatant to the PCR tube. So then the sample preparation is complete. So click the orange tray to continue the part which is the PCR amplification. For the PCR, so we have to uh, take the PCR master mix, adding the master mix solution to the PCR tubes. Then we have to take the positive control DNA, adding the control DNA to the positive control PCR reaction tube. Then for the negative control reaction, you will add water. Click the blue cap deionized water bottle to continue. So the click the PCR machine to open and loading the compartment and load the PCR tubes. Now we have to run the PCR amplification. So click and above to pause or replay the animation. So now you could see the animation over here. Okay, this is the PCR amplification process. So now click the orange tray to continue the PCR product purification. For the product purification, we set up the micro concentrator column. Click the yellow capped buffer bottle to continue. Adding buffer solution to the column. Then discarding the tip and click on the orange tray. Adding PCR product to the column. Discarding the tip. Now the tubes containing the PCR sample, positive control and negative control, will be put on ice. So now loading the column into the centrifuge, then adding the counterbalance to the centrifuge, then centrifuging. DNA is now trapped in the column. Now we need to loosen it into a new tube. So click the orange to continue. So inverted column. So now is adding buffer solution to the inverted column. After that, discard the tip. The first collection tube is no longer needed. So click the orange to continue. So discarding the first collection tube. So now the column into the centrifuge. Now adding the counterbalance and click the centrifuge to continue centrifuging. Afterward, DNA has now been collected in the collection tube. The column is no longer needed and will be discarded. So now the PCR product will be purified. Then we going to the DNA sequencing. Now we will dilute the PCR product to increase the volume. So discarding the tip, and the green and blue strips tubes are already contain PCR reaction mixtures in them. So click the stripe tubes to continue. So now we adding the purified PCR product to the strip tubes. Now the strip tubes are ready for PCR amplification. Click the PCR machine to continue. Now the PCR amplification is running. So now this is the animation of the uh, DNA sequencing. Okay, the sequencing preparation is complete. So now we're going to do the DNA sequencing. Now the DNA sequencing machine to open the doors and load the. So now you already put so the inner gray door. So this is the animation of the DNA sequencing. So now we have to complete the DNA sequencing. Now we go into the sequencing analysis. For the identify the bacteria. For that, I choose Bartonella hensin and answer recorded. The click sample install of the sequence analysis on the sample. Yes. So the identification is correct. So the Bartholomew Hensel, the sample as was obtained from tissue and fluid aspirated from lymph node of patient complaining of a fever, fatigue, and swollen lymph glands. So my results is correct. So now we already done the simulation. Thank you, madam.